Welcome, 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 everybody. Good morning, good evening, good day. Why I say good morning? Because I'm located out in California right now, and it's about 6:30 in the morning. So if I look a little rough, nothing that makeup couldn't fix this morning, but even that couldn't fix there. So welcome. Thank you for joining. I know everyone on this call is probably busy, but this topic is super important, and my colleague and I, Candy, are excited to talk to you about it today. So today we're going to be covering regulatory essentials for pipetting and liquid handling workflows. My name is Nick Anea. I am the product manager for Artel by Nova Biomedical, which we just acquired as part of Advanced Instruments. I am the product manager for Artel. Have a bunch of field experience. I was an application scientist for around 5.5 years, and I've been with the company for about 7 years. I'm going to throw it over to my colleague Candy real quick to introduce herself, and then we'll get the show on the road.
Awesome. Thank you so much, Nick. And thank you everyone for taking time out of your busy schedule to join us. As Nick mentioned, my name is Candy Gilman. I'm the Education and Training Manager for the Artel portfolio line of Advanced Instruments, now Nova. We're very, very excited to have you join us today. With regards to my background, I've been with the company. I just finished my 17th year, so I'm embarking on my 18th year. I started when I was 4, if those of you are trying to figure out the math and my age. Just kidding. And my background is certainly with a lot with regards to pipetting. So I've certified hundreds, dare I say, maybe thousands of analysts and scientists on pipetting technique, worked with a lot of our customers to implement in-house pipette quality management programs that include calibration, pipette repair and maintenance, as well as operator assessment. And between Nick and I, we know quite a bit about this topic that we're getting ready to start this journey on. So a couple of housekeeping things. First things first, we would love to hear your questions. So if you have any questions as Nick and I proceed through this presentation, please place them into the chat. And I want you to know that even though you're placing your questions into the chat box, they're not going to be answered on the fly live, if you will. We do have a question and answer segment at the end of our presentation. And at that point, we'll address any questions that you're placing into the chat box. And if for some odd reason there's so much interest in so many questions that we're not able to answer them all, no worries. We will answer them, and we'll send the answers to those questions along with a recording of this webinar to you after we wrap up today. So again, if you have any questions, we definitely want to hear from you. You will be sent a recording, and we've got a lot of great information to share with you. So I'm going to volley this back over to Nick to get us started.
Thank you, MC with the Moses Candy. Let's go on. So for the agenda today, I'm going to be covering 2 sections. The first 2 sections, so overview of regulatory hierarchy, and then ISO volumetric program requirements. And then I'm going to shoot it back over to Candy to cover regulatory applications for liquid handling and just how we can maybe help you guys from our technology perspective. We're not here to sell anyone on anything today. We just want to give you all the tools necessary for you to understand some requirements in your field around liquid handling and see if we can help in any way by providing you with some of the tools that we have at Artel also. Over to the next slide, please. So why is this important? Why is liquid handling important, right? Why is intercomparability of liquid handling performance between instrumentation important? Think about it, guys. Liquid handling is essential to all laboratory applications. As a young biochemist just out of school, I never thought about that, right? It was, I'm designing this IVD assay. I trust my pipette. I trust when I move it over to an automation engineer that volume is going to be delivered correctly. But if that's off, that calls into question everything that I'm doing. And it also calls into question some of the regulatory guidelines I'm adhering to. So it's really important to establish a quality pipette system across, especially for an international company that's multinational across multiple sites. So an example we have here, it really is imperative for this company that has, pipettes in Raleigh, North Carolina, and Brussels, and then Shanghai to all be on the same page, all be using the same set of quality management tools. Next slide, please. So getting back into this as well, liquid handling quality assurance and standardization are critical, right? Pipettes are essential probably to everything that everyone does on this call when it comes to the laboratory. Pipettes are the start of an assay. They're the start of an experiments. They're the start of a scientific process. Now, operators as well. Operators are the ones that are normally controlling these pipettes. Even, even automated pipettes, they're controlled at the end of the day by operators. So also standardizing our operators and making sure that they can deliver volume correctly and standardize correctly as well. Automated liquid handling, eventually as you scale, if you are running a lot, you will scale to an automated liquid handler. So how do you check the performance of those things? How do we optimize those liquid handlers with the different reagents that we're working with to then deliver the volumes that we need? And then really just understanding the requirements and standards in the different areas and functions that we play at. And hopefully today, we're going to be going over a little bit of that and getting everyone to think about this a little bit more. And hopefully giving you a little bit of a guide to then look at the standards in your area, or maybe if you know the standards, to read into them a little bit more with a little bit more of an understanding. Next slide, please. So Candy and I are going to try to make this as interactive and as fun as possible. So what I want everyone to kind of think about with this question is, think about your laboratory right now. What does your laboratory program fall under? A, ISO recommendations, whatever your SOP says, is there something that your SOP says? My external vendor services take care of all of that. I never think about that. Or other standards or guidelines that are not mentioned in this question. So take a minute. Ben threw up a poll here. You could actually put your answer in and it'll pop up in the chat, and we can kind of take a view of how everyone answers this question here as well. Give everyone a second to do that. Cool. I see we're funneling in over here, and it looks like a lot of people are ISO recommendations. Wow. The majority, around 68%, 66%, whatever my SOP says, that's a good one. As a bench scientist in my previous life, that's exactly what I adhered to. I had no clue what the standards ultimately were. I just followed my SOP. Some have external vendor services and other standards / guidelines, but a majority on this call adhere to ISO recommendations, which is really, really good, and a topic we're going to be covering today. So great. Wonderful. I appreciate everyone who volunteered in the poll. It's good to see those answers and know what baseline we're working with. Now, I also want to just throw out a question here. And we're not going to answer this question until the end. I want everyone to kind of think of this throughout the call. What's the difference between calibration and a verification / routine test? Now, why I'm asking this question is I hope the contents of the day will get us through this. It's common in this industry for me to see people get these wrong, the difference between calibration and verification. So I just want to throw that out there for everyone to kind of think about. And hopefully we could help answer that question along the way. And by the end of this call, you guys will be more expert than me or Candy at answering this question as well. Next slide, please. All right. So let's get into the meat and potatoes of this presentation. We, in our minds, the way that we structured this is a regulatory hierarchy. We like to call it a little bit of pyramid. We're going to go into segments of the pyramid individually. But at the top of the pyramid, we're going to start out with laws and regulations. In the middle of the pyramid, we're going to move towards program requirements. And lastly, we're going to talk about consensus standards and guidelines. So we're going to go into them with a lot of nuance here and talking a little bit more about them. But we'll take each section and break it down because it starts from a very narrow scope and broadens out as we move down. So hopefully you guys will get a flavor of what each means and how they kind of intersect together to create this whole pyramid. Next slide, please. So, like I promised, we're going to start out with laws and regulations. Slide. So when we talk about laws and regulations, especially in the US, we think about the FDA. Now, I think in individual countries, there's their own version of the FDA. For the FDA for the US, we have some very seminal acts that came into place in 1938. That was basically when the FDA was first established with the Food and Drug Cosmetic Act. In 2011, we have the Food and Safety Modernization Act. But under those guidelines as well, there's clinical Clinical Laboratory Improvement Amendments that are part of CLIA in that FDA Code of Federal Regulations. But in your specific countries, there are bodies like the FDA in the US that are these overarching institutions that are really seminal to federal law and regulations. You cannot, hopefully in your countries, just as well in the US, you cannot not obey the rules and laws of the FDA. And these are some seminal moments of the FDA when it comes to regulations. But in your own countries, I'm sure they mirror this specifically as well. Next slide, please. Under the FDA as well, there's specific codes of federal regulation. There's all of these CFR guidelines as well. Same thing from a European perspective. There's the annexes that we adhere to in those specific, there's a bunch of these specific codes, but these are codes for federal regulations, and each one of these codes and part numbers cover a different thing. You'll hear 21 CFR Part 11 a lot. That's electronic records and signatures. There's 21 CFR Part 58, which covers good laboratory practices. But all of these codes essentially, and there's thousands of them, stand for a different thing and a different regulation that you have to adhere to be compliant with the Federal Drug Agency as well. Next slide, please. So once again, what's paramount specifically in the biopharma industry, and this even holds its weight globally. In the US specifically, we have 21 CFR Part 11. This is the FDA guideline that defines the criteria for electronic records and electronic record signatures to be trustworthy and equivalent to paper records. So if you've ever seen audit trails or e-signatures, that's adherence to 21 CFR Part 11. In the European Union specifically, it's Annex 11, which outlines the European Union guidelines for the use of computerized systems that are related to industries to comply with GMP or good manufacturing practices as well. These two regulations kind of mirror one another. They're just called different things based on where you're at as well. There are other codes globally that probably adhere to it. I'm sure there's some in our Asia region as well that adhere to 21 CFR Part 11, or it might just be a copy of 21 CFR Part 11 or EU Annex 11 as well. So think about this, especially when it comes to software for liquid handling, verification, or calibration. Oftentimes, the way that the data is processed, especially if it's through a computer, they have to adhere to one of these standards ultimately. Next slide, please. Okay, so we kind of went over laws and regulations. So that's kind of the big bucket that we have to adhere to. It's federal law in order, to adhere to it. Now, narrowing it down and being a little bit more nuanced, there's specific program requirements that are relevant to industries that you pertain to as well. So we're going to get into that a little bit currently. Next slide, please. So in the U.S., once again, because we're a U.S.-based company, but these hold true. We're going to get into some of these international bodies as well. We have a few different bodies that a lot of our customers like to adhere to. There's CAP, which stands for the College of American Pathologists, and CAP accreditation is discipline specific to a checklist. So in order to be CAP accredited, you have to follow a specific set of requirements. And there's also CAP ISO 15189 accreditation. So that's a lot of numbers. We're going to go over that in a second. So don't worry about what that specific ISO standard is. So a lot of our clinical laboratories have to be under a CAP compliance ultimately. So they strive and are accredited to CAP and must follow these checklists as well. When it comes to, okay, we go back for a second as well. Sorry, I think we went ahead. There's what's called American Society of Crime Laboratory Directors. That's more of the laboratory of the criminology type forensics area, and that's ASCLD ISO 17025 accreditation. Once again, ISO 17025 is a huge calibration standard, which this body makes sure that if you're accredited to, you have to be ultimately under. And lastly, the biggest one, especially in the US, is the American Association for Laboratory Accreditation, HULA for short. And in order to be accredited to HULA, you have to adhere to ISO 17025 and ISO 15189. That was a lot, right? There's a lot of ISOs that are thrown in there. We're going to break that down as we move forward and to say what these specific ISO standards actually are. But in general, if you adhere to a specific requirement, there's different areas that you have to adhere to. So like I said, CAP in the US, HULA, those are bigger ones in the US that a lot of our customers adhere to. Next slide, please. So I said before, HULA is an American organization. How does that apply internationally? I know most people on this call are international here, but in the international community, we have what's called ILAC. ILAC stands for International Laboratory Accreditation Cooperation. So this really ensures that global acceptance of test and calibration results through mutual recognition among accreditation bodies. So what does that mean, right? And you can hit the next slide for me as well, Candy, over here. This means essentially that HULA is accredited with ILAC, which also has a bunch of these other accreditation bodies that are mutually recognized. So currently within ILAC, there's about 116 accreditation bodies, which spans a lot of different, probably about 116 countries as well. Just because it is not an ILAC accreditation, ILAC accredited or part of that accreditation body, does not mean that it's not mutually recognized as well. So I want to think about this and make sure that if you don't see that whatever country you may be in is part of ILAC, it still may be recognized internationally as part of ILAC as well. It's just not listed in the accreditation bodies that are mutually recognized. So what does this ultimately look like? Next slide, please, Candy. Different accreditation bodies all over. So I think we have friends from Italy, Spain, Portugal here, Norway, maybe Turkey, all of the different accreditation bodies, right? So in Italy, Accredia, in Portugal, IPAC, all the way down to France being COFRAC or in Germany being DEX. All these different accreditation bodies are in mutual recognition with ILAC. So if there's a standard that in the US under HULA, these accrediting bodies could recognize HULA as well because HULA is part of ILAC as well. So these are some of our European accreditation bodies. Next slide, please, Candy. Not to forget about some of our Middle Eastern and Asian accreditation bodies as well, and with Egypt with EGAT, and then all the way down to South Korea with COLAS, Japan with JAB, China with CNAS as well. All of these, these are just a few, but this kind of shows you all of these accreditation bodies that are a part of ILAC in this mutually recognized standards. Next slide, please. Okay, so we just went over kind of laws and regulations, program requirements, but I think this is kind of where why everyone's here today is consensus standards and guidelines. After seeing everyone saying they adhere to ISO recommendations, this is really the meat and potatoes of what I want everyone to think about and take away today. So next slide. Okay, I guess we gave the answer away here too, but this is a good teaching moment. ISO 17025 provides detailed instructions for calibrating volumetric devices. So I know Ben might put the poll on the screen here. We already kind of gave the answer away. This is false. Why? I hear 17025 a lot, and you'll hear it in your laboratories. You'll probably hear it with your QA and QC departments. We adhere to 17025, or we follow 17025. 17025 is just a huge standard that outlines calibration. Doesn't matter what you're calibrating specifically, it just outlines all of calibration. There's specific standards that actually go over volumetric calibration. So when you hear 17025, that's not the standard for all for pipette or volumetric or liquid handling calibration or automated liquid handler calibration. This is just the overall standard for calibration in general. So important to think about that because I hear that confused all the time. What does 17025 tell us? That's not specifically for volumetric verification or calibration. Next slide, please. So there's consensus standards that are mostly voluntary, but some are requirements. So I heard ISO earlier. ISO stands for the International Organization for Standardization. So that's applied globally. There's ASTM International. that's also it's an American. It started in America, but it also spans internationally. And then there's the Clinical Laboratory and Standards Institute, which is CLSI as well. So I would think most everyone on this call adheres to one of these standards, unless you're kind of at R&D or something and you're not adhering to one of these standards. But most of these standards within a regulated environment are pretty streamlined, whether it's ISO, ASTM, or CLIS globally as well. Next slide, please. Okay. This is the big one. If you don't remember anything that Candy and I say today, I want you to take away this one slide. So ISO 17025 and ISO 15189. That's the what? That's a set of general requirements for calibration that calibration providers must follow to be deemed competent and suitable to an accreditation body. So this could be pipettes, this could be car tires, this could be that refrigerator that's sitting in your laboratory, or that minus 70 degrees Fahrenheit freezer. It's ISO 17025 and 15189 is the what. Around the volumetric standards, specifically for pipettes, it's ISO 8655. That is a very detailed set of standards and methods that control pipette calibration providers must follow to correctly and accurately calibrate a piston pipette. And more so around automated liquid handling, it's ISO 23783, which is the procedure for volumetric performance to meet the requirements of the standard for automated liquid handling systems. So once again, 17025 and 15189 are the what. And the how do I actually do this, is 8655 and 23783. So once again, if you take away nothing from this presentation, I want you to remember those two specific things, because oftentimes people will say I follow ISO 17025 or ISO 15189. That's just the what. How do I actually do this? The granularity is in ISO 8655 and ISO 23783 when it comes to automated liquid handling. Next slide, please. And then we have this nice little table as well that kind of shows you where these standards originate from and kind of the evolution. You can see these are these volumetric motor pipettes are under ISO 4787, but more so probably for the purposes of this audience, both piston operated single and multi-channel pipettes are under ISO 8655, and then under automated liquid handlers that are acoustic or positive displacement or air displacement are under ISO 23783. And there's a couple other standards for those specifically as well. But those specifically are how you calibrate and verify. Remember, nothing from today, ISO 855 for manual piston operated pipettes, and then ISO 23783 for automated liquid handling. Next slide, please. And lastly, and this is what Ben is going to provide you with our follow-up email. This is a huge chart. We try to break down different clinical or different applications and what regulatory requirements based on our pyramid they would fall under. So after this call today, our follow-up email to you is going to be a copy of this presentation for you guys to think about. And then also this table, it'll be nice and cleaned up for everyone to take a look at and to see general guideline to see what we fall into. Different pyramid of what we talked about with regulations today. Next slide, please. So I'm going to hand it over to my colleague Candy. So we kind of went over the theory here. Candy's going to really get into applicationally what this ultimately looks like and walk us through why this is important within the laboratory setting. So take it away, Candy.
Awesome. Thank you so much, Nick. And it never ceases to amaze me that Nick wakes up and he's got all of this energy. So kudos to you, Nick, for having all that energy. I think your computer was starting to say that's enough talking because your audio started to clip in and out, but hopefully everyone can hear me okay now. All right, so now that Nick has really kind of laid the foundation and the groundwork for what are these federal mandates versus these consensus standards, let's really talk about how do you apply all the details that are contained in some of those. So the one thing that I want to start with is a disclaimer that this presentation is not intended to replace the actual actual purchase and adherence to any of the standards and guidelines that we talk about. What we've done is we've really kind of tidied everything up nicely to give you a really high-level, broad overview, but it doesn't take the place of actually purchasing the standard and pouring through it line by line and making sure that everything is in play. So let's go ahead and go to the next slide. So first things first, as we start thinking about a volumetric calibration or verification program, there's a lot of different puzzle pieces that go into that. Things like what volumes do you intend to test at? What replicates? How many replicates per volume do you intend to take? If you have a multi-channel device, which channels are you intending to test? Just a little bit of inside secret for you. If there's multiple channels, they should all be tested, right? What type of test method are you going to be using? Are you going to be using gravimetry? Are you going to be using titration or dual diphotometry? The tolerances, what are you considering pass or fail? And the frequency, how often are you going to perform your calibration or verification of your volumetric devices in your laboratory? And you'll see at the very center of this puzzle, operator training is right in the center, because the fact is you can define that program as detailed as you'd like. But if the person who's performing the pipette calibration doesn't know how to operate the pipette well, and you don't have their training documented and you're not assessing them on a consistent basis, then your program and all those puzzle pieces, they really start to fall apart and they start to split apart. So as we continue exploring how do these standards relate to these specific parts of the puzzle, you'll get a little bit more sense and idea of what we're looking at. So specifically, as Nick mentioned, he had said, if you take away anything from this presentation, just know that your ISO 8655 series, that deals with piston-operated volumetric apparatus. Now, that in short, ISO calls that POVA, so I promise I'm not going to quiz you on that, but POVAs are not just pipettes. They're also considered burettes, diluters, dispenses, syringes, all fall under that piston-operated volumetric apparatus category. 23783 deals with the automated side of the house. So you move all of the manual pipetting over to an automated robot, and we were really excited when 23783 came out in 2022 because there wasn't really a standard globally that addressed if you have these robots in-house, how should you be verifying their performance? So we were really excited when that standard came out. And then as Nick had mentioned earlier as well, the CLSI standard QMS 23 specifically, which talks about general laboratory equipment performance, qualification, use, and maintenance. A lot of words. Pipettes fall within that standard as well. So let's go ahead and explore a little bit deeper. Specifically, when we talk about the ISO standards, I'm going to be giving you some details about Part 6, 7, and 8, and then also some information about the entire 23783 series and information about pipettes with relation to QMS 23. So if you take a look at this particular chart, down on the left-hand side are those pieces of the puzzle that I just mentioned to you. What test method do you intend to use when you're checking the volumes of these devices? How frequently are you going to check their volumes? If you have multiple channels, which channels do you intend to test? What volume settings will be tested? How many replicates per volume setting? What do your tolerances look like? And then, oh, by the way, don't forget about the operator training and assessment. So we're specifically, as I mentioned, going to be talking about ISO 8655, ISO 23783, and that CLSI QMS 23. So let's go ahead and take another look. specifically at the 8655 series. So when we think about POVA, piston-operated volumetric apparatus, we're talking about manual pipettes or semi-automatic pipettes. Those of you that might use those wonderful electronic pipettes where you can aspirate 1,000 microliters and do 10 shots of 100, those multi-dispensers, they are just wonderful devices, right? Those fall under this set of standards as well. So specifically, when we think about Part 7 and Part 8, Part 7 references a ratiometric photometric method, which our systems fall under. That also mentions a gravimetric method in Parts 6 and 7 as well. Specifically, what they talk about with regards to frequency is that it's really up to you sometimes as far as how frequently you're testing these devices. Now, oftentimes, people just want a very prescriptive roadmap to say, tell me what I need to do. I'll write my SOPs and I'll do it. And so ISO 8655, it really gives you some prescriptive methods, specifically in the reference measurement standards that are very, very, very detailed. But then Part 7 becomes a little bit less stringent, and it's a little bit more applicable for your everyday lab. We're going to talk about that. But the nice thing about it is that you can find some autonomy, and then you can also find something that's very prescriptive and very detailed that you should follow. With regards to channels, ISO 8655 indicates every channel should be tested. And then with regards to volume settings, what volumes do you intend to test? If you have an adjustable device, especially an adjustable pipette, calibrations should involve testing at the low volume, the mid volume, and the high volume. But if you're just performing routine tests, those volume settings, you could have fewer volume settings that you're testing. Maybe you decide to test just at the low and the high volume. And of course, what you don't see if you do that is you won't be able to get to a linearity answer, if you will. But those routine tests are really just meant to be a quick check of that device. Now, when we think about replicates per volume testing, or per volume setting, I should say, calibration, a full calibration means 10 replicates at each volume. So if you're calibrating an adjustable volume pipette at 3 volumes, 10 times 3 is 30. That means 30 replicates to perform a calibration according to 8655. Routine tests, you can have a minimum of 4 volumes. So again, routine tests are meant to just be that quick check. Make sure and answer the question of, is this device still within its calibration? Maybe somebody dropped the pipette and you just want to check it quickly, or maybe something got aspirated into the body of the pipette and you want to make sure that it hasn't been contaminated, make sure that it still is A-OK. Now, with regards to tolerances, I have a specific slide that we're going to talk about in tolerances, but ISO provides a little bit of autonomy. They say, listen, laboratory, you can certainly define your own tolerances based on that device's process requirements, or ISO 8655 Part 2 provides some permissible error tables for some very distinct classifications of devices, air displacement pipettes, etc. Or you could use the manufacturer specifications. I'm going to talk about this, as I mentioned, in a subsequent slide, because there's some things that you should consider when you think about tolerances. And then last but not least, ISO 8655 Part 10 came out in 2024. That's all about user qualification, user training, and assessment. That particular standard indicates that any pipette user or any user of a POVA should be trained, that's important of course, should be consistently assessed and monitored at a minimum of every 12 months. So those operators that are using those pipettes, maybe your pipettes are being calibrated and they perform beautifully after they're calibrated, but if you place them into the hands of an operator who doesn't know how to use them in the most ideal fashion, you're not going to get the best results you can. So making sure that you have those records on hand is important. Now let's kind of swap over to the automation side of the house. So ISO 23783, as I mentioned, came out in 2022. Before that, ISO definitely recognized that there was a need to have some type of standard in the world with regards to these automated liquid handling devices. So IWA 15 came out in 2015, and then we finally got that formal standard in 23783 in 2022. So with regards to the test method options for automated liquid handlers, there's 8 different methods that are included in 23783, including the dual die ratiometric photometric method, which is where our systems come into play. Now, with regards to frequency, it's a little bit different for automated liquid handlers. It really depends on it's a risk-based evaluation. So how often you test that automated liquid handler? It could be a quarterly, it could be every 6 months, could be annually. What's the acceptable amount of risk if it's not delivering the correct volume? But then maybe you also have some user requirements or even manufacturer suggestions that you follow. So lots of autonomy with 23783. That's intentional. It's really kind of the first version, if you will, with regards to automated liquid handlers. I think that we'll probably see as the years go by that ISO might put a little bit more detail around the recommendations and requirements in that standard. The channels to be tested, much like 8655, every channel. Okay, volume settings, much like 8655. So if you're looking to claim linearity, testing 3 volumes over the testing range of that automated liquid handler and its head that you're testing with it. Testing 3 volumes will give you that ability to calculate linearity across the device. The replicates look a lot like the pipette calibration side of the world. For calibration of those devices, 10, a minimum of 10 replicates. For routine tests, a minimum of 4. Now here's where things get a little bit interesting. So with regards to tolerances for automated liquid handlers, there aren't any specifications in 23783 because it really depends. There's a lot of things that go into 23783, including the fact that you can use different fluid types apart from water. Maybe you choose to optimize and test glycerol or ethanol or something like that. So with that, right, comes a bit more autonomy and certainly a lot more ambiguity, if you will, with regards to tolerances. And then, of course, operator training and assessment is not applicable because now you're just essentially programming software and telling the robot to do what you want it to do. So let's kind of round it full circle with CLSI QMS 23. So again, that's the standard that a lot of our clinical customers will follow and adhere to, if you will. So they mention a couple of different test methods in their standards. Photometry, a photometric method, as well as a gravimetric method is allowed. They also have a mention about frequency. You see that those 2 words, risk-based, are also mentioned in QMS 23, but they recommend a quarterly calibration, which is interesting, right? So that's a bit more frequent than what ISO recommends. But they also indicate that maybe it's more frequent than quarterly for critical pipettes and less frequent for less critical pipettes. So again, they're really providing the lab some autonomy to think about how are your pipettes used and what's really important. They follow the same thought process as ISO as far as the number of channels to be tested, every one of them. And then they also follow similar thoughts with regards to the volume testing. 3 volumes are recommended, a minimum of 2 volumes for calibration. The replicates, minimum of 10, and then tolerances, very, very similar to ISO. However, they have some very specific permissible errors when it comes to systematic error and random error. So plus or minus 2% for systematic error or accuracy, as some of you might refer to that, and then 1% of full scale for random error or imprecision. So they have some distinct suggestions if you decide to not implement a process or lab requirement. And then with regards to operator training and assessment, performance qualification checklists require proof of training. And that's really as much as they say. They don't give you much detail about that, but really where all the detail comes into play is the ISO standards. So let's talk a little bit more about tolerances. This tends to be a bit of a hot topic for us. So let's take, for example, you can see on this particular slide I have depicted a Rainin LTS pipette. It's a 20 to 200 microliter single channel device. Now, with regards to ISO, as well as CLSI, they have some recommendations, but let's take a look at this very first column, which is the manufacturer's specification. So Rainin has said that if you're testing at 200 microliters, that relative inaccuracies plus or minus 0.8%, imprecision is 0.15. Those are really tight tolerances. When you take a look at ISO 8655 Part 2's table of permissible errors, they align nicely, right, with regards to inaccuracy. They also indicate plus or minus 0.8%, but their imprecision is a little bit broader than Rainin's, so they indicate imprecision of 0.3%. And then we've got the CLSI standard. They indicate relative inaccuracy of plus or minus 2%, imprecision of 1%. So then where does that leave you in the decision making, right? Because all of the standards indicate that you could also implement your own process tolerances when it comes to what's considered passing or failing for this one pipette. So for example, if the laboratory process indicates that this pipette can deliver plus or minus 5% from an accuracy standpoint, maybe you don't need to meet that Rainin spec or that ISO spec of 0.8%. If that range during its true process is plus or minus 5%, 0.8 is probably a little too tight. Maybe you decide to have your calibration follow plus or minus 3%. So it's well within that plus or minus 5% process tolerance, but certainly a little bit tighter. So just definitely some things to think about, right? As you think about, am I going to use the pipette's manufacturer specification? Am I going to use ISO 8655's permissible error table? Am I going to follow what CLSI recommends? Or should I really be looking at what is required of this device during the process itself? Now, there's another bit that comes in with ISO 8655, and that's the environmental side of things. Now, you can see here I've listed ISO 8655 Part 6 and Part 8 across the top. These are the reference measurement procedures for performing a gravimetric calibration, as well as a photometric calibration. Now, when we think about reference measurement procedure, that's a very stringent procedure that the laboratory has to follow from A to Z. You can't pick different things that you want to adhere to. If you say that you are following 8655 Part 6 or 8655 Part 8, that means you're adhering to all of it, not just some of it. So the environmental impact comes into play especially for air displacement pipettes. So Part 6 and Part 8 indicate that your laboratory temperature should be 20 degrees Celsius plus or minus 3 degrees with a max variation of 0.5 degrees Celsius per hour. So if you have a highly uncontrolled laboratory environment, maybe you're the first person to enter the lab in the morning, it's very cold. And then as your colleagues enter the lab, the temperature increases and you can see that temperature differing once you have 20 bodies in the lab versus no bodies in the lab. How fast is it differing? That can affect how these pipettes perform. Humidity is another column or another row that we want to take a peek at. Air displacement pipettes are very dependent on humidity. So according to ISO 8655 Part 6 and Part 8, they advise that your laboratory should have a constant humidity of 45 to 80%. So you walk into the laboratory and you feel like you're in a sauna, right? Because you can feel the moisture in the air. Air displacement pipettes love having a moist environment to work in. Now, if you take a look at the ISO 8655 Part 7 column, that's the alternative measurement procedure. Now, gravimetry and photometry are also mentioned in Part 7, but there's a little bit less stringent of a process. Specifically, they don't indicate in Part 7, ISO doesn't indicate in Part 7 the specific temperature your lab needs to adhere to, although they do mention that it shouldn't be shifting more than 0.5 degrees Celsius per hour. But they don't specifically recommend humidity levels of what your lab adheres to. And then when we get over to the clinical standards, CLSI QMS 23, they just indicate that it's method dependent. So a lot of ambiguity, but also a lot of autonomy. So if you kind of take that glass half full mentality, that means that you've got the ability to define it yourself. Now here's what we know. ISO 8655 Part 2 has an annex in the back of it, and it shows you how much variation that the environment could have on an air displacement pipette. You could have potentially a 3.5% impact on your volume strictly coming from the environment of your lab. If it's really, really dry in the laboratory, if the temperature is fluctuating a lot, you could see that type of impact on your volume. And so that's definitely something that we want to consider for sure. All right, so we've got another poll. I'm going to have Ben open the poll up for us. And so the poll question is, if you were following ISO 8655 to calibrate your pipettes, how many pipette tips would you use in a single volume 10 data point calibration? So go ahead and take a moment and answer the poll question to the best of your ability. And then we're going to talk about it, the answer, in just a moment. All right, we've got a lot of answers coming in. Thank you. All right, those of you that might have fallen asleep, we've got a poll question on the screen. If you're following ISO 8655 to calibrate your pipettes, how many tips would you use in a single volume 10 data point calibration? So I'll give you a few more seconds to submit your answers for those of you that want to submit an answer. All right, so you have to forgive my line of sight. I'm looking over at the chat to see what our poll answers are coming through as. It looks like a single pipette's definitely a popular choice for many of you. All right, let's go ahead and take a look and see what the answer is. And the answer is 2. So those of you that answered 2 tips, we're going to talk about why is it 2 tips. And for those of you that answered something other than 2, you might be kind of sitting there with a puzzled look on your face thinking, what in the world does that mean? Well, we're going to go ahead and talk about that. All right, so not only does the environment come into play when it comes to following ISO standards for pipette calibrations and verifications, but also the method comes into play, specifically how are you operating the pipette. So if you take a look across the top, I've got the reference measurement procedure for 8655 Part 6, which is for gravimetric testing, Part 8, which is for photometric testing, Part 7, that's that alternative measurement procedure, which follows a lot of what Parts 6 and 8 mention, but has a little bit more flexibility for your everyday lab. And then rounding the end, we've got QMS 23. So the very first thing or technique that we see here is the number of tips that are used. And according to ISO 8655, Part 6, 7, and 8, they recommend that you change tips at a rate of n equals 5. So that means after 5 replicates, you're changing your tip. So those of you that answered 2, 5 times 2, right? Because we've got 10 data points that we're taking, you're absolutely correct. You can give yourself a little pat on the back. All right, so let's go down a little bit further just real quick and let's take a look at the rest of these techniques. These are not all inclusive, but these are some of the ones that I wanted to specifically call out to you. So the second row that you see is the orientation during aspiration. When you're aspirating, the pipette should be held completely vertical. If you're going through the lab and you're seeing your pipette operators holding the pipettes at angles, they're not getting the correct volume. So that's specifically called out. Pre-wetting is another technique that's called out in all of the ISO standards, and I'm going to talk a little bit about pre-wetting in just a moment. Pipette tip immersion depth is something that's also called out in the ISO standards. So they recommend 1 to 6 millimeters dependent on the volume. Now, you might be surprised, 6 millimeters is not very deep. And so oftentimes you'll see folks just kind of taking their pipette tip and just placing it so deep into the solution. It's not great, right? It definitely causes some differences in results. Pausing after aspiration. That is 1 or 3 seconds, depending on the volume that you're working with. And then dispensing, the ISO standards recommend that you dispense at an angle. So this particular webinar is not about pipetting technique, but it is about standards. But specifically what I want to call out is talking about this pre-wetting technique. So let's quickly take a peek. So what I'm going to do is I'm going to play this video, and what I'd like for you folks to do, and this is not a poll per se, but what I'd like for you to do is watch the video. And if you are performing pre-wetting in your laboratories, is this the pre-wetting technique that you see that's being used or is written into your procedures? And so if you are so inclined, you can answer yes or no in the chat. So let me go ahead and play that again for you. This is where the tip is placed into the solution and it stays in the solution and the plunger is actuated up and down, up and down. All right, so again, if this is the pre-wetting technique that you utilize within your laboratory and it looks familiar, you can go ahead and say yes or no, or you can choose to plead the fifth and not answer. It's totally your choice. All right, I see some answers coming through. All right, wonderful. Thank you for those of you that have contributed your answers. All right. So here's what we know about this technique. This is what we call the imposter technique, right? And so there's a reason why this is called the imposter technique, right? The intention for properly pre-wetting a pipette is for you to place your pipette tip into the solution and to pull your aspirate and then remove your tip from the solution and then place your tip against the side of the wall according to ISO 8655 recommendations and drag that tip up the side wall. And that is one single pre-wet. Now, when it comes to ISO 8655, they recommend that you do that 5 times. And then they recommend that you take 5 replicates. And then they recommend that you put on a new tip and you pre-wet 5 times and take 5 replicates. That's what your 10 data point single volume calibration process would look like. So some of you, I hope, have a good takeaway from this particular slide. So let's talk a little bit more about 8655 and the training requirements. So Part 10 states that the specifications in 8655 are to be carried out by trained personnel who use, test, or calibrate PROVA, and that their competence should be demonstrated and documented. Now down here on the bottom, again, as I mentioned with my introduction, I've trained hundreds, if not thousands, of analysts on pipetting technique. So what you see on this left-hand side scatter plot chart is true data of pre-training data of what we've gathered and as found of what we've gathered. And then post-training data, once we educate analysts on how to properly pipette and why it's so important and giving them one-on-one coaching. So you can definitely see that, with a little bit of attention and a little bit of training, that you can see data is drastically improved. And ISO is pretty well documented about the impact of the operator's technique. So depending on how your operators are using pipettes, they could contribute up to 8% influence on volumes. So if you add that to the 3.5% influence from the environment, you're now up to an 11.5% influence on your volume. Right. And so you can see how the error compounds very quickly and why this stuff is so important when it comes to all of that the maintenance and the structure of your liquid volume verification in-house. All right. So how do we help? So we help in several different ways, right? So we have 2 different flagship instruments, if you will, the RTEL PCS, which stands for pipette calibration. It's really intended for single channel devices, but also for operator training and qualification. The RTEL MVS system, which stands for Multi-Channel Verification System. Again, that deals with automated liquid handlers with regards to ISO 23783, the calibration of multi-channel pipettes, as well as operator training and qualification. And then as Nick mentioned, Nick talked about 21 CFR Part 11. The software that uses those two systems has the ability to facilitate conformance to 21 CFR Part 11. So there's audit trail as well as electronic signature functions and tasks within the system, and then operator certification and training and qualification. So that's our certificate programs that we have. Now, shameless plug, those of you that are interested in coming to the state of Maine in the United States, I have a pipette quality management certification course. It's 2 days in October, and October 16th and 17th, where we'll train you how to implement an in-house pipette quality management program, including pipette repair and maintenance, pipette technique, etc. So if you're interested in coming to Maine and having some lobster, please let us know. So again, as we mentioned, we're just going to kind of come full circle. What parts of 8655 are relevant to our instruments and pipette certifications? All of the 8655 parts, 1, 2, 3, 4, 5, and 9, as well as for 6, 7, and 8, like all kinds of different parts, right? So all of the parts that we've been talking about all along, but then also including part 10, which has to do with operators. And then 23783, again, that has to do with liquid handlers. All of those parts of the ISO standards are relevant to our MVS system, our RTEL portfolio. And then, as I mentioned earlier, the RTELware application that we use for the PCS and MVS systems has what we call a compliance suite add-on, and that enables you to comply or help comply to 21 CFR Part 11. So provides you with audit trail, electronic signatures, and a few other things with regards to electronic record compliance. All right, so Nick, I'm going to have you come back in if you'd like, if your audio is working okay, and if you want to round us out here at the end.
Sure.
Awesome.
I am back. Hopefully you guys, everyone can hear me well enough here as well. So getting back kind of into our technology as a whole, the biggest thing that we hear a lot is traceability, right? The MVS and the PCS are both traceable to international system standard or units, NIST USA and NPL-UK. So when it comes to traceability and having a source for traceability, whether that be gravimetry or other standards, we're there and we got you when it comes to traceability. Next slide, please. And then also, our own labs, anything that comes out of our labs are compliant with ISO 17025, and we have our A2LA accredited, which means we're also ILAC accredited as well. So there's peace of mind when you choose RTEL and you choose our systems. As kind of Candy walked you through before, all these standards are very complex and hard. They're very nuanced. I think our systems and our technology really helps you because the way we build them is with all of these standards in mind, so you're not really thinking about it. They're already there, and they conform to probably whatever regulatory body or whatever standard you're ultimately under. Next slide, please. And once again, we always will give you documentation with our ISO 17025 accreditation, which shows the measurements that are taken and how we adhere to them for both of our PCS and our MVS systems. So, you know, if that's ever a concern, we always have our certifications present and ready for you or your end customers. Next slide, please. Okay, so I asked the question at the start of this, what is the difference between calibration and verification? And hopefully we guided you through this in a very nuanced way. But calibration is a traceable measurement consisting of a defined number of replicates with a specified tolerance limits and may or may not consist of physical adjustments. And that's the key. I think most people think calibration involves me physically changing something. That is so far from the actual fact, it's unbelievable. So really, calibration just means that we are within the tolerance limits that we set, and that we're showing those tolerance limits in a uniform way. Versus verification, normally occurs between calibrations to conform to that the device still meets its specified tolerance limits and may have a lower number of replicates than a calibration. So Candy kind of went over in a very detailed way of the difference between how to calibrate and how to verify. Next slide, please. So that was a lot in a very finite amount of time. I know we have 2 minutes left, but congratulations, everybody. That was even for me and Candy, this is a lot that we covered in a very short amount of time. So we thank you all for being here. Hopefully it was very impactful and enlightening to you. We have maybe a couple minutes for Q&A. So if anyone has any questions, please feel free to put them in the chat. If not, we will follow up. You feel free to answer questions through email, and we will get back to you as quick as we possibly can.